Welcome to another video tutorial on bacterial growth curve and factors affecting bacterial growth. Bacterial growth. Bacterial growth is an increase in the number of bacterial cells rather than the increase in their cell size. So yes, generally when we speak about growth, so growth is the increase in the cell size or increase in the size of the organism, right? But here bacterial growth refers to the increase in the number of cells. Okay, like for example, if there is 2 bacteria, so 2 becoming 4 and 4 becoming 8. So like this, the increase in number is what we call it as bacterial growth. Okay, and then bacteria are unicellular organisms that tend to reproduce asexually by the means of binary fission. So yes, we all know that bacteria is a unicellular organism. Okay, and they reproduce, or I can tell they produce 2 daughter cells by means of an asexual method which is called as binary fission. Okay, as you can see here, this is a method in which one single parent cell becomes 2 daughter cells. Okay, and here in binary fission, as you can see, so there is a cell over here and with its nucleoid. Okay, so first the nucleoid splits into 2, so it undergoes replication and the nucleoid splits into 2, and then here over here the cytoplasmic membrane actually elongates the cell, and then cross walls will be formed in between and 2 daughter cells will be formed finally. Okay, so this is how binary fission will happen. Okay, and then this is how the single cell will split into 2. Now, the growth of these bacterial cells takes place in an exponential manner, that is, one cell divides into 2, then 4, then 8, 16, 32 and so on. So yes, sir. Now this division of the cell takes place in a very systematic manner, or I can tell in a very exponential manner. So now as you can see here, one cell gave rise to 2 daughter cells, and now this one cell will be able to give 2 cells again, and this cell will be able to give 2 cells again. So like this, now one cell will be giving 2 cells, and again out of 2 4 will be formed, and again from 4 8 will be formed, from 8 to 16, 16 to 32 and so on. So like this, in an order, in an exponential manner, the division will happen. And then the time taken for a bacterial cell to double is called generation time. The generation time varies among different species of bacteria based on the environmental conditions they grow in. Yes. So now the time taken for a single cell to become 2 is called as generation time. Okay. So, and it varies in different species of bacteria. Okay, there are certain species of bacteria which takes minutes. So only within a few minutes, one cell becomes 2. Whereas there are certain other bacteria, so which takes hours together. Okay, like that. Clostridium perfringens is the fastest growing bacteria that has a generation time of 10 minutes, while Escherichia coli has a doubling time of 20 minutes. So as you can see here, this Clostridium perfringens, it's one of the fastest growing. Just within 10 minutes, it can split one cell into 2. Okay, the same thing when you take in Escherichia coli, it takes about 20 minutes, double the time. Okay, and so when you take Mycobacterium tuberculosis is one of the slowest growing bacteria, taking about 12 to 16 hours to double. So this takes about 12 to 16 hours. So just imagine. Okay, so it can be minutes, and for others bacteria it can be even hours. Then bacterial growth curve. In a closed system with enough nutrients, a bacteria shows a predictable growth pattern, that is a bacterial growth curve. So yes, in a suitable lab conditions, that is in a closed system, no, so when you give enough nutrients and allow the bacteria to grow, it shows a definite pattern of growth, and that definite pattern of growth is what you call it as bacterial growth curve. Okay, so that is nutrients and nothing but they are food for the bacteria to survive. Okay, when you give enough nutrients, enough food for the growth of bacteria, and when you allow the bacteria to grow, what all happens, no, that happens in a particular pattern. So that is the bacterial growth curve here. So you can see the growth curve in the picture over here. Okay, so and it has four different phases, that is lag phase, log phase or exponential phase, stationary phase and death phase. Okay, so as you can see in the picture here, so this is the lag phase and there is a sudden increase which is called as log phase or exponential phase, and then the graph becomes stationary. Okay, so there is no increase or decrease, it becomes stationary, so that's called as stationary phase, and finally the graph drops down, which is called as death or decline phase. Now we'll study them one by one. First is lag phase. The bacteria upon introduction into the nutrient medium takes some time to adapt to the new environment. There is no increase in cell number but only increase in size. Bacteria adapt to their new environment, building up the necessary enzymes and metabolic intermediates for the next phase. Yes, lag phase is also called as adjustment phase. Okay, when you put a new bacteria into a nutrient medium, the place will be new for them, the nutrients will be new for them, right? So it needs a adjustment phase. Okay, so that adjustment phase is a lag phase. Here bacteria will actually not increase in number, they don't reproduce, they first adjust themselves. Okay, they themselves grow in the size here and they prepare. So they prepare, like they build up all the necessary enzymes and all the metabolic intermediates and all those things they will synthesize and they'll prepare for the next phase. Okay, hence you call it as preparation phase or adjustment phase. And then next is log phase or exponential phase. The log phase is also known as exponential phase. During this phase, bacteria experience maximal growth rate and the population doubles at a constant rate. Bacteria begin to multiply and their number increase exponentially with time. The exponential phase is marked by a steep upward slope on the growth curve graph. Yes, that is, now as we saw in the lag phase, the bacteria got adjusted. It also synthesized the necessary enzymes required for the upcoming phases, right? So now in this phase called log phase, no, they utilize the maximum amount of nutrients and they start doubling. Okay, and so they show maximal growth here. They utilize as much nutrients as possible and they keep on dividing and they show maximal growth in this phase, and that's why you can see a upward slope here. There is a sudden rise and a steep upward slope can be seen in the graph over here. Okay, and factors such as favorable environmental conditions and enough nutrients contribute to the high metabolic activity and proliferation of bacterial cells during this stage. So yes, now apart from the nutrients, no, so favorable environmental conditions also matter for them to double up. Okay, so because of the suitable environmental conditions and plenty of nutrients, no, they show maximum growth in this phase. And then the growth rate slows down when toxin substances start to accumulate. So yes, now what happens is, now the when the bacteria is utilized as much nutrients as it can, no, the nutrient level will start depleting. Now it becomes lesser and lesser when there is no enough nutrients available and the number of bacteria is too much, no, certain bacteria will start to die. Okay, when some bacteria dies, no, it accumulates some toxin substances into the environment, and when toxin substances starts accumulating, no, the growth rate also slows down. Okay, then stationary phase. The stationary phase is the third phase of bacterial cell growth and is often referred to as state of equilibrium. The number of new cells created equals the number of dying cells. Yes. Okay, now the bacteria is utilized as much nutrients as it can and the nutrient level is come down now. As a result, what is happening? The cells are dying and toxins are getting accumulated. And particularly in this stage, in the stationary phase, what happens is the number of new cells created will be equal to the number of the cells dying. Okay, because nutrients is not supplied to the all the bacteria over there. So hence half of the bacteria is dying and half of it are created. And hence you call it as a state of equilibrium. So both are equal here. And that's why the graph is stationary over here. And then the bacterial growth and multiplication slows down as nutrients are exhausted and toxin products accumulates. So yes, now as and so the nutrients is coming down and down and down, the multiplication, the bacterial growth also becomes less. Okay, and toxin products will get accumulated more and more. Then finally, decline phase or the death phase. The death phase of the bacterial growth is a final phase of the bacterial growth. It's when the rate of cell death exceeds the rate of cell division in a bacterial cell or culture. Yes, that is the death phase or decline phase is the last phase. So where almost all the nutrients have been utilized and there is no nutrients available for the bacteria. And as a result, what happens? Almost all the cells will start dying and you can see a slope in the curve. So there is a drop down in the curve. Okay, and so the rate of the death here exceeds, it becomes more than the cells which are created. Okay, so hence we call it as death phase or decline phase. So these are the different stages of bacterial growth curve. Okay, in which they get adapted. Lag phase is where they get adapted and they prepare for the division, and then in the log phase they show maximum growth. They utilize almost all the nutrients here. Okay, once when the nutrients are utilized, so the number of death and the number of the cells created will become equal, and that's called as stationary phase. And when the nutrients become very less, the death rate will increase, and that's called as death or decline phase. So this is all about bacterial growth curve. Then okay, next is factors affecting the bacterial growth. So first is nutrition. So yes, nutrition plays one of the important role in growth of bacteria. Okay, so as we all know that when there is adequate supply of nutrients, so when there is enough nutrients, no, the bacteria utilizes the nutrients and they divide, and so they show maximum growth rates. Okay, but when there is limited supply or when the nutrients are getting depleted, so it affects the bacterial growth becomes slower and they reach decline phase. Yes, bacteria need nutrients such as carbon, nitrogen and essential minerals to reproduce and move. Adequate nutrients support bacterial growth and limited nutrient can lead to slower growth rates. Yes, so mainly bacteria requires carbon source, nitrogen source and essential minerals to reproduce and to move. Okay, when there is enough nutrients they grow and they show maximal growth rates, and when there is limited supply of nutrients, no, the growth rates will be slower. That's about nutrition. And then next is pH levels. pH stands for potence of hydrogen. And here speaking about pH, no, pH actually varies for different organisms. Okay, so and depending on what type of organism you need to grow, so based on that the pH level also varies, right? And then in environment with neutral pH, bacteria growth is fast. Yes, but most of the bacteria prefer neutral pH, that is pH 7 is most preferable, and the bacteria shows fastest growth in neutral pH itself. Okay, but there are some other bacteria which can grow in basic pH, which will grow in acidic pH. Okay, like See this. Most pathogenic bacteria grow between pH 7.2 to 7.6. Yes, pathogenic bacteria is the bacteria which causes diseases. Okay, so this bacteria grows between the pH 7.2 to 7.6, a slightly basic pH. Whereas there are some other bacteria which grows in acidic pH also, like lactobacilli. Okay, so lactobacilli chooses pH below 4, so it grows in acidic pH also, but most of the bacteria will choose neutral pH for their growth. And then oxygen. Bacteria on the basis of their oxygen requirements can be classified broadly into aerobic and anaerobic bacteria. Yes, um, now as we discussed in the previous videos and all, not all bacteria require oxygen for their survival. There are many bacteria which require oxygen for survival, and there are some other bacteria which do not require oxygen for survival. Okay, so based on that we can divide them into aerobic and anaerobic. As the name tells here, aerobic means these are the organisms which require air or oxygen for their growth, and anaerobic, as the name tells, anaerobic, so these are the bacteria which do not require air or oxygen for their growth. Okay, we'll study them one by one here. So first is aerobic bacteria. They require oxygen for their growth. Okay, so they require only in the presence of oxygen they will grow. So hence we call it as aerobic bacteria. Then, so now aerobes are of three types. First is obligate aerobes, which can grow only in the presence of oxygen. So yes, obligate aerobes are strict aerobes. Okay, they can grow and survive only in the presence of oxygen. Without oxygen, they cannot grow and survive. Example is Pseudomonas aeruginosa. And the second type is facultative aerobes. So which are ordinary aerobes but can also grow without oxygen. Yes, so these are aerobes. Okay, so they require oxygen for their growth and they're happy if they have oxygen, they grow and survive. But they can also tolerate anaerobic conditions. They can also grow and survive in anaerobic conditions. Example for that is E. coli. Okay, and most of the pathogenic bacteria are facultative aerobes. Yes, most of the disease causing bacteria are facultative aerobes. Then the third type is microaerophilic bacteria. As the name tells here, micro means small, aerophilic is air. Okay, that is less amount of air. So those bacteria that can grow in the presence of low oxygen and in the presence of low concentration of carbon dioxide. Yeah, so that is even if the amount of oxygen is less, they can grow and survive very easily. So such bacteria is microaerophilic bacteria. Example is Campylobacter jejuni. Yeah, and the second type here is anaerobic bacteria, that is they do not require oxygen for their growth and survival. Example, and the first type is obligate anaerobes. Okay, so bacteria that can grow only in the absence of oxygen. Yes, so obligate anaerobes are again strict anaerobes. They do not require oxygen for their growth. Okay, even a small amount of oxygen can kill them. So such organisms are called as obligate anaerobes. Example is Clostridium botulinum, Clostridium tetani, etc. Okay, and the second type is facultative anaerobes. These bacteria can grow in both the presence and absence of oxygen. Yes, so they can grow in the presence as well as absence of oxygen. Okay, example is Streptococcus and Staphylococcus. And then third is aerotolerant anaerobes. As the name tells here, aerotolerant, that means they can tolerate air. Okay, so these are anaerobes which can tolerate air. See, so these bacteria can tolerate oxygen for a limited time but do not use it for growth. Yes, so these are anaerobes. They do not require oxygen for their growth, but they can tolerate oxygen for a limited period of time. Okay, so but they do not grow. They survive with oxygen in a limited period of time. So example is lactobacillus. Okay, and the next important factor is temperature. The optimal temperature for most of the pathogenic bacteria is 37 degrees Celsius. Yes, for most of the disease causing bacteria, So the optimal temperature, that is a temperature which is required for their growth and survival, is 37 degree Celsius. Okay, but it is not that all the bacteria will grow in the same temperature. There are many bacteria which can even grow in the coldest of the environment, in the coldest temperature and even in the hottest temperature. Okay, so the optimal temperature, however, is variable depending on their temperature range. Growth of bacteria is grouped as following. Yes, so based on the temperature requirements, we can divide them into three types. First is psychrophiles. These bacteria are cold loving microbes that grow within a temperature range of 0 to 20 degree. Yes, so they are cold loving microbes. So they grow and survive in the cold environment. So in the temperature range of about 0 to 20 degree. Okay, and most of the soil and water saprophytes belong to this group. Okay, saprophytes are the bacteria which depend on the dead and decaying organic matter. Okay, so saprophytes which are living in soil and water all belong to this group called as psychrophils. Example is Arthrobacter. And the second type is mesophils. As the name tells here, meso means moderate temperature, middle. It's not too hot or cold. Okay, so these are moderate temperature loving microbes that grow between 25 degree to 40 degree. Yes, so they can grow in the moderate temperature range between 25 to 40. Okay, and most of the pathogenic bacteria belong to this group. Yes, most of the disease causing bacteria has optimal temperature which falls between this range only. Okay, that is 25 to 40. Example is E. coli. And the third group is thermophiles. So these are heat loving microbes. So they can grow at a high temperature range of 55 to 80 degrees. Yes. So they grow in a hottest environment, that is they are heat loving microbes. Okay, and they grow in the temperature range of about 55 to 80 degree. Okay, example is the cyclobacillus. Okay, this is about temperature. And then next factor is light radiation. So yes, so some of the bacteria require light, so and they derive their energy from light, and some other bacteria will derive their energy from chemicals. Okay, see now depending on the source of energy, bacteria make use of, they may be classified as phototropes. As the name tells you, photo means light. So bacteria deriving energy from sunlight. So these are the bacteria which derive energy from the sunlight. Okay, and then chemotropes. So chemotropes, bacteria deriving energy from the chemical sources. Yes, as the name tells here, these are the bacteria which derives energy from the chemical sources. Okay, then okay, so next factor is carbon dioxide. Okay, yeah, carbon dioxide also plays a very crucial role in the growth and survival of microorganisms. Okay, especially bacteria. And so while we speak about bacteria, no, all bacteria requires some amount of carbon dioxide for their growth. Okay, so the minimum amount can be 0.04%, whereas the maximum amount can be 5 to 10%. Okay, that is there are bacteria which can survive in the minimal amount of carbon dioxide like 0.04% to the maximum, that is they require higher concentration like 5 to 10% also. Okay, now so based on that, the organism that require higher amounts of carbon dioxide for their growth are called capnophilic bacteria. Yes, so the organism which require higher concentrations of carbon dioxide for their growth, no, they are called as capnophilic bacteria. So how much they require? They require around 5 to 10% of carbon dioxide for their growth. Okay, and 15% oxygen. Fine. And then in candle jar, 3% carbon dioxide can be achieved. Yes, in candle jar itself we can achieve 3% carbon dioxide. An example for them is Haemophilus influenza and Brucella abortus. Okay, so next is osmotic pressure. Next and the last factor is osmotic pressure. Okay, now what's osmotic pressure? So I can tell it is a pressure exerted on the cell membrane, okay, due to the differences in the solute concentration between the interior and exterior environment. That means now I can tell here, see this is a cell, as you can see in the picture, this is a cell, and as you can see here, the solute concentration is different outside as well as inside. Okay, when there is difference in the solute concentration, no, so there is a force exerted on this cytoplasmic membrane. Okay, so the water from outside or the solution from outside exerts a pressure on the cytoplasmic membrane, so which is called as osmotic pressure. Okay, now microbes obtain almost all the nutrients in solution from surrounding water. Yes, so almost all of the nutrients which they require and the minerals which they require, they get it from outside only. They get it through water. Okay, and hence factors such as osmotic pressure and salt concentration of the solution affects the growth of bacteria. So yes, so hence the osmotic pressure, of course the pressure will be exerted on the cytoplasmic membrane, so the osmotic pressure and the salt concentration, so everything will affect the growth of the bacteria. Okay, and the bacteria, by virtue of mechanical strength of their cell wall, are able to withstand a wide range of external osmotic variations. Yes, so because the cytoplasmic membrane, no, they have a mechanical strength, so they can withstand all of this osmotic pressure, and it is required for the bacteria to survive and grow. Okay, and organisms requiring high osmotic pressure are called as osmophilic bacteria, while there are certain other bacteria which require high osmotic pressure. Okay, such bacteria we call it as osmophilic bacteria. Okay, then